Alright everyone, so today is the last unit within energy systems, and today we'll be going over actually the 3 energy systems that we need to know and a little bit about nutrition as well. We're going to start off by looking at a cell. So looking at the cell, we've got quite a few different labels on here, but luckily for you guys within the IB, you only need to know 7 of them, and hopefully some of these are quite familiar to you from biology. So we've got the nucleus, we've got the cytoplasm, the mitochondria. Remember, you can see it's written there as mitochondrion. So mitochondrion is 1 singular and mitochondria is plural. Lysosomes, the rough endoplasmic reticulum, ribosomes, and the Golgi apparatus. So we're going to run through each of these now. You need to be able to label them if you saw a picture of a cell. So we'll talk about their location, where you can find them, and just a brief description of their function. So the first one, the nucleus, very easy to find. It's the command center, normally a big circle that is in the center or slightly off center within the cell. It holds all of the cell's genetic information, so the DNA, and it tells the cell what to do. It tells it when to grow, when to make proteins, or when to divide if necessary as well. Our next cell part is the cytoplasm. So the cytoplasm, I've put a label in there and you can see that arrow in red and it's kind of pointing towards nothing. That's because the cytoplasm is the thick jelly that sort of fills the cell. It's where all the different organelles, all the different parts of the cell sort of float around, and it's where a lot of important chemical reactions happen to keep the cell working. Importantly, anaerobic metabolism, which we'll get quite deep into today, occurs within the cytoplasm of the cell. We'll hear more about that a little bit later. Then we've got the rough endoplasmic reticulum. So the rough endoplasmic reticulum is normally always surrounding the nucleus. So you can see here it's that blue structure surrounding the nucleus. So if you're looking to label it on a diagram, find the nucleus, know that the rough endoplasmic reticulum will be around it. Importantly, also, the rough endoplasmic reticulum is covered in ribosomes, and that's what gives it that rough look. So when you look at the blue here, you can see those little red dots. Those are the ribosomes that are giving it that look. Within the rough endoplasmic reticulum, in terms of its function, it's a bit like a protein workshop. It's where proteins are made and then sort of folded into the right shapes in order to leave the cell or go to the cell membrane. So it's sort of creating and synthesizing and processing the proteins that are coming, that are in the cell. All right, we've got the ribosomes. So remember, the ribosomes are those little dots that are on the rough endoplasmic reticulum. They are the sort of tiny factories that make the proteins, which then go into the rough endoplasmic reticulum to be processed and synthesized. Ribosomes make a lot of different proteins that are really important for different functions in the body. So some examples include insulin. So we've spoken a lot about insulin within this unit. Insulin is made by the ribosomes, and remember, it helps to regulate our blood sugar levels. Within the ribosomes, we also make hemoglobin. So hemoglobin we've spoken about as well. It's a protein in our erythrocytes, our red blood cells, that carries oxygen from the lungs to the rest of the body. Then we've got the Golgi apparatus. So the Golgi apparatus is the green in the image on your screen. Think of it as like the post office in the cell. It takes the proteins and the lipids and whatever's made in the cell and then sort of sorts and packages them off to be sent wherever they're needed. The lysosomes, so they're a little bit harder to see on this diagram here. It's sort of that perfectly blue little circle that the arrow is pointing towards. But the lysosome is like the cell's recycling center. So it breaks down waste and old cell parts and anything harmful that could be in the cell so that the cell stays clean and healthy. And then finally, we have the mitochondria, or mitochondrion, if we're just talking about one. And we all know mitochondria, it's the powerhouse of the cell. But what does that actually mean? So the mitochondria takes in nutrients and turns them into energy, so ATP. They also help to control the cell's lifestyle, but mainly we're talking about, especially when we're referring to the powerhouse of the cell, we're talking about taking in nutrients and converting it into ATP. And very important to remember, aerobic metabolism occurs here. So when we're using oxygen to create that ATP, it's all happening within the mitochondria. When it's anaerobic, that was back in the cytoplasm. Again, we'll go more into that within this unit. So looking specifically at a mitochondria, we need to know a few important features on this organelle as well. So specific parts of the mitochondria. Firstly, the outer smooth membrane. So this membrane sort of encloses the mitochondria, sort of like creates the barrier between the mitochondria and the cytoplasm that it's floating around in, and it allows for little molecules to pass into the mitochondria when they need to. Then we've got the cristae. So the cristae is the folds on the inner membrane of the mitochondria. So you can see there those yellow folds within that mitochondria. That's the cristae. Firstly, they increase the surface area within the mitochondria for chemical reactions to occur. And the important chemical reaction that occurs here is the electron transport chain. This is a really important part of aerobic metabolism that occurs in the cristae, the electron transport chain. Again, we're going to talk more about that soon enough. And then finally, we've got the inner matrix, which is the space enclosed by sort of the inner membrane of the mitochondria. It's sort of like the free space within the mitochondria. It contains different enzymes and DNA, or mitochondrial DNA specifically, and ribosomes. But most importantly, this is where the Krebs cycle occurs. Again, the Krebs cycle, just like the electron transport chain, is a very important part of aerobic metabolism. So remember, anaerobic metabolism occurs in the cytoplasm. Aerobic metabolism occurs in the mitochondria, with different parts of aerobic metabolism taking place within different areas of the mitochondria. Electron transport chain in the cristae and the Krebs cycle within the inner matrix. Okay, so a definition for you now, cell respiration. So cellular or cell respiration is the metabolic process where cells convert nutrients, primarily glucose, into energy or ATP. So we've got two primary types of cell respiration that we've already mentioned: anaerobic metabolism and aerobic metabolism. Let's talk about aerobic metabolism first. So aerobic metabolism occurs when there's oxygen present within the cell. So cells utilize oxygen to break down glucose, and we know that that occurs within the mitochondria. When we use our oxidative system or our aerobic metabolism, it produces quite a substantial amount of energy. So with one glucose molecule, we're getting approximately 34 ATP. If you look in different areas, if you're studying for this course on YouTube, you might see different numbers. Some sources say 36 or 38, but we're going to go with 34. But just know that it's an approximate number. In terms of byproducts, so if I'm creating 34 ATP out of one glucose, are there any byproducts? Is there anything else that I'm creating along with that? And with aerobic metabolism, we, as well as ATP, we create carbon dioxide, we create water, and we create heat. And these are sort of expelled through respiration or breathing, especially the carbon dioxide and the water. Our second form of cell respiration is anaerobic metabolism, and this occurs in the absence of oxygen, so without oxygen. So when we don't have oxygen within the cell's going to use different systems to create energy. These are known as the phosphagen system or the glycolytic system. So two different systems within our aerobic metabolism. And we know already that this happens within the cytoplasm of the cell. Now, anaerobic metabolism produces much less energy. One glucose creates between 1 to 2 ATP molecules. So the reason it's 1 to 2, just to point that out, is the phosphagen system is 1 glucose, 1 ATP. The glycolytic system is 1 glucose, 2 ATP. Again, we're going to see more about that later. In terms of byproducts, we have lactic acid. So I'm sure you've all heard of lactic acid before, that lactic acid burn that you get after you run, 200 meters as fast as you can, and you feel like your legs and your bottom are like pulsating and it's so painful, you feel like you're going to throw up. That's from that lactic acid and that increase of hydrogen within the muscles, within the cells, which creates this acidity within the cells. So it's decreasing the pH within the cells. In terms of conditions for these two different types of metabolism, for anaerobic metabolism, we're talking about intense exercise, short bursts of activity, or low oxygen environments. So altitude might increase our anaerobic metabolism because we just don't have that oxygen coming to the cells anymore. For aerobic metabolism, this one's really important because people tend to associate aerobic metabolism with just like moderate, steady state exercise, like, you go for a jog, that's aerobic. But also remember that during rest and everyday activities, your body's relying on aerobic metabolism. Your body doesn't need to be producing that much energy when you're resting, so it's very easy for the body to maintain the oxygen required for that level of ATP. We'll get more into that as well. But rest, light to moderate exercise, and everyday activities very much relies on our aerobic metabolism. So just day to day, sitting here watching this lecture right now, you're probably using your aerobic metabolism. Okay, so throughout this unit, we've discussed various pathways to glucose. How do we get glucose into the cell? Firstly, we said glucose in the bloodstream. So if I've eaten a high carbohydrate meal, I've got glucose in the bloodstream, it can enter the cell with the help of insulin. Secondly, we said glycogenolysis. This is the process where we retrieve glucose from that stored glycogen. So we've got that stored glycogen in our muscles and in our liver. That's broken down into glucose through glycogenolysis. And then we've spoken about ATP and what that means. So within this first sort of system here, glucose to ATP, we're either going to go through one of two sort of pathways. It's either going to be aerobic, where there's oxygen, or it's going to be anaerobic, where there's no oxygen. The only thing that we sort of haven't covered at all is how do we get from ATP to energy? So we understand that the formation of ATP from glucose is influenced by either the presence or the absence of oxygen. We haven't yet covered what ATP is exactly and how it functions as sort of like the body's energy currency. So you can see the molecule of ATP in the middle there. We've got adenosine triphosphate. So ATP, adenosine triphosphate. Okay, so firstly, let's look at that molecule. Within a single ATP molecule, we have one adenosine, which is that big blue blob on your screen, and we have 3 phosphates. So you can see 3 little P's coming off of that adenosine. I can't say that word. Okay, easy to remember because triphosphate, right? Tri means three phosphates. Now, there is a very important process called hydrolysis, which is where we will use water and an enzyme called ATPase, or sometimes people call it ATPase. So water plus this particular enzyme cause that third phosphate to break off and produce energy. So that's actually how ATP works to create energy. It's the breakdown, it's the hydrolysis of this ATP molecule, which then combines with water. The last phosphate splits away and energy releases. Once the energy releases, we've got that one single lonely phosphate group floating away. And we've got now adenosine diphosphate. So notice how it's changed from tri to diphosphate, di meaning two. So we're only left with two little phosphate groups attached to that adenosine, and we've got one independent phosphate group. Okay, so I just want to mention this before we continue on and look deeper at the three different energy systems. So energy systems within the body are often referred to by various names, and depending on where you look for revision or what you've said in class, you might have used different names. Now the IB goes with phosphagen, glycolytic, and oxidative. So that's how I'm going to talk about these three energy systems for the rest of this lesson. But just notice that, for example, the phosphagen system is also known as the ATP-PC system, the ATP-PCR system, the ATP-CP system, so they've just switched the P and the C around there, and the anaerobic alactic system. The glycolytic system, you might have heard of it as anaerobic glycolysis or the lactic acid system. And then oxidative, also referred to as aerobic glycolysis, oxidative glycolysis, aerobic metabolism. There's so many different ways that people mention these three systems. So you can always refer back here if you're confused. If you read something and you don't know what they're talking about, refer back here and you can figure out which of the three systems it is. So let's have a look at our three systems now. Firstly, phosphagen. Phosphagen is anaerobic. So for a weightlifter, for example, the phosphagen system is essential. It provides rapid bursts of energy for like explosive movements during short, intense lifts or short, intense bursts of activity. The next one is glycolytic, which is also anaerobic, so without oxygen. A 400-meter runner will rely on the glycolytic system. It supports high-intensity efforts, but with sort of a limited duration. And the glycolytic system is the one that produces lactate. So that lactate burn is primarily coming from usage of that glycolytic system. And then the last one we have is oxidative. This is our aerobic system, so we're using oxygen within this system. Think like a 10K runner, they're going to rely predominantly on the aerobic system, which is able to produce energy over a prolonged period and really supports like endurance sports. So we're going to look at these three systems today, and oftentimes people like to refer to the three systems or sort of use an analogy for the three systems as technology. So some people say that the phosphagen system is like the iPhone. The iPhone provides quick, immediate access to like texting or calls. It's very rapid, but it drains quickly if we use it too much. So like our iPhone dies. So we don't want to use that for long, complex tasks. The next one, the glycolytic system, people say it's like an iPad. It handles longer tasks like watching a movie or playing games or whatever you're doing on your iPad. It's a bit more durable than the iPhone, but eventually it will lose charge as well and fatigue over time. And then the last one, the oxidative, people refer to that one as the MacBook or the laptop, designed for long-term use, such as working on like a big project or editing videos. Represents that aerobic system that's a bit slower to start, right? You got to turn the laptop on. Maybe it takes a while to turn on, but it provides sustained, efficient energy for long duration activities. So the iPhone, phosphagen, provides quick but short-lived energy. The iPad, glycolytic, provides moderate energy for longer periods. And then the MacBook supports prolonged, steady energy. So that sort of illustrates those three energy systems if it helps you to think of them as Apple products. Okay, the phosphagen system. The phosphagen, the phosphagen system consists of ATP and creatine phosphate, also known as phosphocreatine or PCR. It's referring to the same thing. So when phosphocreatine is broken down, it releases a phosphate and energy, which is then used to regenerate ATP from ADP through a process called phosphorylation. And we're going to talk about that in a second. So in terms of products, one phosphocreatine can create one ATP, and there's no byproducts. In the phosphagen system, again, it operates anaerobically, so it doesn't require oxygen, and it's the predominant energy system during the first 5 seconds of exercise, regardless of the intensity. This system is the primary source of energy when you first start exercising. So we actually have some stored ATP within the body, but that stored ATP level lasts like a couple of seconds, maybe 3 to 5 seconds. And then the PCR, or the phosphocreatine, helps to buffer the drop in ATP for an additional 5 to 8 seconds. So together, the stored ATP plus the phosphocreatine helping to sort of buffer that drop, this system supports exercise of around 10 seconds duration. Okay, so let's see how this actually works. So we've got a stored ATP that we've used as energy, and we've turned that into ADP. So you can see within this first diagram here, I've used that ATP, I've turned it into energy. Now I'm left with adenosine diphosphate and a random phosphate group. So what's going to happen is that phosphocreatine is going to interact with something called creatine kinase, and it's going to donate its phosphate group to the ADP, so to the adenosine diphosphate. So the creatine kinase will break that bond between the phosphocreatine, and the leftover phosphate group will join back up and create adenosine triphosphate. So there's been some talk of phosphocreatine, creatine kinase. It might have reminded you of a supplement called creatine that a lot of people take. So in terms of the function of creatine, if you're taking it as a supplement, you're mixing it with water and drinking it, creatine supplementation helps to boost the phosphocreatine levels within the body and enhances the phosphagen system's efficiency. So with higher phosphocreatine levels, we're able to slightly longer maintain a maximum effort and improve performance, but it's really only important for activities like sprinting, jumping, heavy lifting. It's not going to help us over time. It's within that 10-second time period, remember. The more phosphocreatine, or the more creatine we have as well, we can have a quicker recovery between short bursts of intense activity. So think like team sports now, or interval training, or even at the gym. You want to recover quicker between sets in order to produce the same effort. And it actually doesn't take phosphocreatine to regenerate. Doesn't take very long for phosphocreatine to regenerate. So about 80 to 90% of your phosphocreatine stores are restored within about 1 to 2 minutes rest, and I think it's completely replenished within 3 to 5 minutes. That is why a team sport athlete like soccer uses predominantly this phosphagen system, because they have these really short, intense bursts of activity, and then quite long rest sometimes. And with that rest, they're able to replenish that phosphocreatine and continue to use that when they have their next burst of energy or running. Same thing with resistance training. So if you're in the gym doing really, really heavy weights, you'll have a very long rest period. And the reason that you have a very long rest period is in order to replenish those phosphocreatine stores, which are really going to help you produce that energy that you need within that short time frame of doing your weightlifting, right? So creatine supplementation can help to boost phosphocreatine levels, but remember, it's only really good if it's applicable to your sport or whatever you're doing. If you want to lift really heavy weights in the gym or have a really good 100-meter sprint, then it might be a supplement that you consider. If not, then if you're training for endurance, it's probably not going to help you all too much, because it will only help within that first 10 seconds if you're doing prolonged exercise. Okay, the next one is the glycolytic system. So the glycolytic system, remember, it's also known as the lactic acid system, becomes the primary source of ATP once the ATP PCR, or the phosphagen system, has been exhausted. So if we're doing a 400-meter sprint, let's take that as an example. In the 400-meter sprint, the glycolytic system is the primary source of energy. Glucose is broken down to produce ATP in the absence of sufficient oxygen. So this process within the glycolytic system converts one glucose molecule into 2 ATP, and it has a byproduct of lactic acid. So lactic acid begins to accumulate in the muscles. The buildup of lactic acid lowers the pH, which leads to that sensation of muscle fatigue and burn that really, really hurts. The duration of this energy system is the predominant energy system for maximal intensity exercise, lasting 30 seconds to 2 minutes. So that is our glycolytic system, the overview. Let's look at how it actually works. So the glycolytic system breaks down glucose, and we've seen this little glucose molecule on the right hand side here too many times by now, but the glycolytic system breaks down glucose through a process called glycolysis to produce ATP. And we've spoken about glycolysis before. Glucose to ATP is glycolysis. The process of glycolysis is super, super complicated. You can see it here. Luckily for us, we don't need to know that. All that we need to know is that glucose is broken down, and you can see the process it's broken down into, but you don't need to worry about the middle part of that graph there. All you need to know is it's broken down into 2 pyruvates and 2 ATPs. So you can see at the bottom of that graph in purple, we've got pyruvate at the bottom, 2 of them, and it's also created 2 ATP. Now, this occurs in the cytoplasm of the cell, and although this system provides energy more slowly than the phosphagen system, it supports activities of a longer duration. So we've already spoken a bit about that. We said the phosphagen system is 10 seconds, whereas this glycolytic system becomes the primary energy source from 30 seconds to 2 minutes. So examples of activities being that 400-meter sprint I keep bringing up, high-intensity interval training, intense cycling or swimming for shorter distances. Now, when there's no oxygen within the cell, pyruvate turns into lactate, and that's how we get that lactic acid. So that's what's occurring there when we have no oxygen within the cell. That glucose is broken down, we get 2 ATPs, but we also get 2 pyruvates. The pyruvates go looking for the oxygen. When there's no oxygen there, the pyruvates turn into lactate or lactic acid. But this can sometimes not be the case let's say the pyruvate finds the oxygen. If there's oxygen in the cell and the pyruvate finds the oxygen, the pyruvate will continue into the oxidative system. So it depends here. This glycolysis, it's at the end when we decide whether we're aerobic or anaerobic. If the pyruvate can't find oxygen, it will turn into lactate and build up that burn within the muscles. If the pyruvate can find oxygen, the pyruvate and the oxygen are going to continue on into the oxidative system. So the oxidative system starts with this glycolysis as well, starts with this process. The only difference is that the pyruvate finds the oxygen, and then we've got a few more processes to go through. So let's see that in a little bit of a graph I've made for you. So at the top, we're looking at anaerobic glycolysis. At the bottom, we're looking at aerobic glycolysis. So firstly, we have our glucose molecule. The glucose molecule goes through the process of glycolysis within the cytoplasm of the cell. This produces 2 ATPs and 2 pyruvates. Those ATPs are used immediately for energy. The pyruvate then turns into lactate when we've got anaerobic glycolysis. Down below, we can see that the pyruvate has found oxygen. It's found O2. So once it's found oxygen, that pyruvate is going to go down to the mitochondria of the cell. And we already know there's 2 things that happen within the mitochondria. There is the electron transport chain and the Krebs cycle. So that's where that pyruvate is going to go in order to create even more energy through aerobic glycolysis. Okay, let's talk about the oxidative system now. We already know a bit about it. So energy sources. The aerobic system can get energy from glucose, from fat, and even from protein in some cases. So the main thing is that it's in the presence of oxygen. The aerobic system is the body's most efficient energy system. It's really important for sustaining prolonged physical activity. If there's enough oxygen, it can use glucose, fats, and proteins as a fuel source. Proteins hardly ever, only when you're starving and the body needs to keep itself alive is when the body will use protein as a primary energy source. Okay, so products. 1 glucose molecule can create 34 ATP. So that's quite an increase from what we've seen before with the phosphagen and the glycolytic system. And it gets even crazier when the oxidative system uses fatty acid. So when we use fat as an energy source, 1 fatty acid can create 1 approximately. So whenever you see that squiggly line, that always means approximately. So that little squiggle line before the 130 is approximately. 1 fatty acid can create approximately 130 ATP molecules. There's a few byproducts. So the breakdown of glucose or fatty acid through the oxidative system results in carbon dioxide, water, and heat. In terms of contribution, the aerobic system is a predominant energy source during rest and also for long duration exercise. It supports that continuous low to moderate intensity activity by providing a steady and sustainable supply of energy. So it becomes a predominant pretty much after 3 minutes. All right, let's look again at how this oxidative system works. We already know a bit about it. The glucose, let's use glucose as an example, and we'll talk about how we use fats later on through the oxidative system. Firstly, we're talking about glucose. So we've got our glucose molecule. Our glucose molecule goes through glycolysis. In glycolysis, I'm creating 2 ATP. I'm also creating 2 pyruvate. There's oxygen in the cell, so the pyruvate is going to connect with that oxygen and travel to the mitochondria. So again, we've got the mitochondria. Once at the mitochondria, the pyruvate turns into something called acetyl-CoA, so acetyl coenzyme A. Acetyl-CoA goes through a process known as the Krebs cycle, where this acetyl-CoA is broken down, producing molecules that carry electrons to the electron transport chain. Within the electron transport chain, electrons are passed along within the mitochondria, generating that 34 ATP. So, very simple explanation of what can be quite complicated. So we know the first few steps. The pyruvate joins with oxygen, goes to the mitochondria. Within the mitochondria, that pyruvate becomes acetyl-CoA. That's just the name of it, just like pyruvate. You don't really need to know any more detail about acetyl-CoA. But we've got to talk about the Krebs cycle. So before I show you this next picture, I just want to warn you that when you go onto YouTube or you're studying the Krebs cycle, you're going to see something that looks like this. Now, for our level, we do not need to know what's going on here at all. It's super, super complicated, but it's this cycle where there's many enzymes and molecules and electrons being created in order to produce more ATP. All you need to know about the Krebs cycle is that it occurs in the inner matrix of the mitochondria. I said that back at the start of this class. What happens in the Krebs cycle is that hydrogen ions liberate, which means release. So hydrogen ions release electrons for the electron transport chain. If you want to know what those ions are and what those electrons are, I've written them down below. It's NAD plus becomes NADH, FAD plus becomes FADH, and then transfers over to the electron transport chain. So the electron transport chain, again, another very complicated looking structure. What we know already is that it occurs in the cristae of the mitochondria. All you need to know about the electron transport chain is that electrons enter the electron transport chain and resynthesize, which means to reform or recreate ATP. And here is where we get that 34 ATP molecules per 1 glucose. Okay, so that is glucose and the oxidative system. Let's talk about fats in the oxidative system. So in aerobic exercise, if we've used all of the glucose available, we're eventually going to move on to using fats for energy. So fats are broken down through a process called beta oxidation. This process releases lots of electrons which move directly to the electron transport chain. So back in the same as the oxidative system with glucose, we eventually get to this electron transport chain. I'm not going to go into the specifics of beta oxidation. All you need to know is that beta oxidation occurs. Many electrons go to the electron transport chain to help produce ATP. And each fatty acid can generate about 130 ATP molecules. It's because fats provide so much energy that they're the main source of fuel for low intensity activities like walking or slow jogging, where we use up all of the glucose for our for oxidation and we turn to fats for energy, and they provide a lot. Okay, so a bit of a recap here. Take your time, take a screenshot of this slide if you want. But the three primary energy systems in the body each play a unique role in producing ATP for different types of physical activity. Firstly, we had the phosphagen system, also known as the ATP-PCR system. This is anaerobic and it operates in the cytoplasm of the cell. It relies on stored ATP and phosphocreatine for quick energy. It provides a rapid but short-lived energy supply, and it produces just one ATP molecule from one phosphocreatine molecule. It's dominant for activities lasting less or up to 10 seconds, such as sprinting or heavy weightlifting. The glycolytic system, also anaerobic. This system breaks down glucose in the cytoplasm of the cell through glycolysis to produce 2 ATP molecules and lactate. It supports moderate to high intensity activities lasting from 30 seconds to 2 minutes. As the activity continues, lactate, this accumulation of lactate can lead to fatigue and that burning sensation. And finally, the oxidative system operates aerobically, so with oxygen, within the mitochondria of the cell. This system uses glucose, fats, or proteins to produce ATP. For glucose, it involves glycolysis to pyruvate. Pyruvate gets converted to acetyl-CoA, which carries on to the Krebs cycle and the electron transport chain, where it generates about 34 ATP molecules. For fats, we've got beta oxidation followed by the electron transport chain, which produces approximately 130 ATP molecules. And that system is predominant for low to moderate intensity activities lasting more than 3 minutes, such as long distance running or even just going for a jog or long walk. Each system is really important for different exercise durations and intensities and ensures the body can efficiently meet its energy demands. So important to remember, you don't just use one system, they all overlap in some way. Now, when you start moving, your stored ATP is the primary source of energy. Then we've got the phosphagen system, which provides immediate energy for short bursts of intense activity, 10 seconds again. So by using stored ATP and phosphocreatine, we're using this phosphagen system. As the activity extends beyond 10 seconds to a few minutes, the glycolytic system takes over and it generates energy through the breakdown of glucose. It's not as efficient and it produces lactic acid. So it's not the best energy system, I guess. There's some downfalls. But for prolonged exercise lasting several minutes to hours, the oxidative system is the dominating system, using oxygen to metabolize carbohydrates and fats, and offering plenty of energy supply for endurance activities. And we can have a look at some different sports here. So again, like I said, the three energy systems don't work independently of one another, they overlap. So notice we've got the sport within the first column, and then look at the next three columns, the titles of the next three columns. These are overlapped energy systems. So we've got the ATP PCR and glycolysis, or the phosphagen and glycolysis. So this is sort of combining the two. Then in the next one, we've got glycolysis and oxidative, combining the two again. And then in the last one, we've got oxidative. So it's not broken up into those three clear energy systems. They've sort of combined them, crossing two different energy systems there. Okay, so let's firstly look at the golf swing, for example. So 95% contribution from the phosphagen and glycolytic systems, 5% glycolytic and oxidative, and 0% oxidative. So if we think about the golf swing, it's a very intense but super short duration activity. So when you think about different sports, always think about intensity and duration. Those are the two variables that are gonna affect the energy system that's being utilized. For skiing, we've got an even cross between phosphagen and glycolytic and oxidative. So you can see it's got 33 for both. I'm not sure if they're referring to just like daily skiing or ski racing. I think this would just be more like regular skiing for a longer duration of time. Soccer is 50% ATP PCR or phosphagen and glycolytic and oxidative is 20%, and then 30% oxidative. So we have a very interesting split here where most of it's actually coming from that phosphagen glycolytic sort of short duration energy system, because like I said before, they've got those short bouts of intense physical activity where they're sprinting for the ball and then resting. That gives the body time to replenish the phosphocreatine that it needs for the phosphagen system so that they continue using that, even though it's a very long duration activity. When we look at the sport itself, if we look at what they're doing within the sport, though, it's those short bursts with a lot of just sort of like gentle running in between, and then big bursts, and then gentle running in between. Field events at the top. So field events, think like your long jump, high jump, javelin, pole vault, very much phosphagen and glycolytic, very much short duration, high intensity, versus running. If we're looking at distance running, that's very orientated towards the oxidative system, so 70% within that oxidative system. So anything distance, anything we're doing for a long time, if we're able to do it for a long time, it means the intensity isn't that high because we're able to sustain it, which means that it's relying on primarily that oxidative system. So duration and intensity are the two things you need to think about. If it's a long duration, it's not going to be a very high intensity. It can't be. We can't sustain the highest possible intensity for a really, really long duration. Okay, so in terms of response to training, so long-term training creates several adaptations in the body's energy systems. It also creates adaptations outside of the body's energy systems, but here if we just look at the energy system adaptations, which enhance performance and endurance, they're listed below. So firstly, we have an increased aerobic and anaerobic enzymes. So the enzymes we really briefly spoke about today, so I think I only mentioned a couple, being creatine kinase, ATPase or ATPase, that were involved in the phosphagen system. But within glycolysis, remember you saw that diagram that had a whole lot of stuff going on, and I said we don't need to talk about that. Those are the enzymes. Within the Krebs cycle, there was a whole heap of stuff going on in the Krebs cycle that we didn't know about. Those are enzymes. So the enzymes are those sort of chemicals or molecules that interact within the process of glycolysis, the Krebs cycle, the electron transport chain. With increased enzymes, we enhance the efficiency of the energy system, allowing for the body to produce energy quicker and sustain exercise for longer periods. The second adaptation is increased use of fats as an energy source. So endurance training in particular improves the body's ability to burn fats for energy. And this is particularly during prolonged, moderate intensity exercise. So this adaptation, if we're able to use fats more to produce energy, we can spare some of those glycogen stores and delay fatigue and allow for longer durations of exercise because we're producing much more energy when we're using fat, remember, 130 ATPs for one fat molecule. With long-term exercise, we can increase our storage of glycogen. So I've mentioned previously in this unit that our glycogen stores are around 500 grams of glycogen, but with regular exercise, especially again endurance training, we increase the muscle's capacity to store glycogen, which is the stored form of glucose. So this provides us with a larger energy reserve for high intensity or prolonged exercise, and again, enhances performance and delays the onset of fatigue. And then finally, we can increase the number of mitochondria that we have within the cell. So we know that mitochondria, they're a powerhouse for aerobic energy production. That's where aerobic energy production occurs. So with consistent aerobic training, the number and the size even of the mitochondria within the cells increases, and that enhances the muscle's ability to generate energy through aerobic pathways, improving our endurance and our overall performance. Okay, we're on to some specific topics that we need to know now. So the first one is excess post-exercise oxygen consumption. So we need to know what that means and what that is. So during intense physical activity, especially at the start of exercise, the body isn't able to supply enough oxygen to the muscles to meet the energy demands, and that creates something called an oxygen deficit. So it occurs at the beginning of exercise until we reach a steady state. So think about if you're going out for a run and you start running, it feels quite hard at the beginning, and then you reach sort of a plateau. You reach this steady state where you're breathing in the amount of oxygen that you need to meet the energy demands. Now, excess post-exercise oxygen consumption, or EPOC, is sometimes referred to as oxygen debt. And this refers to the amount of extra oxygen that the body needs to restore itself back to normal, back to the resting state after strenuous exercise. So after exercise, the body needs to repay this deficit by consuming more oxygen than usual. So during the recovery period, let's say after we've run 5 kilometers, we've stopped, we've reached our 5 kilometer mark, but we're still breathing heavy. We feel like we're dying, we're trying to breathe really hard at the end of that exercise. That's the body repaying this deficit. That's excess post-exercise oxygen consumption. It's the body working to return to its pre-exercise state or its pre-exercise level. So let's see what that looks like. So a few things going on in this graph. We've got this little woman here down in the bottom left corner. At the start of exercise, she's going to start running. Now, the amount of oxygen that she's breathing isn't quite reaching the amount of oxygen that she needs, so that's creating an oxygen deficit that's placed there in the orange. Now she's reached steady state oxygen consumption and she keeps running until she finishes her exercise. Then we have in the light blue epoch, or the oxygen debt. So after exercise, we'll keep breathing really heavy as the body is repaying this deficit by consuming more oxygen than usual during the recovery period. And this epoch, or this increased oxygen intake, helps to replenish energy stores, our ATP and our phosphocreatine. So they're all depleted now. We need to replenish them. It also helps to remove lactic acid from the muscles. It helps to restore the oxygen level in the blood and the tissues, so get our oxygen in the blood back to its normal resting state. And it also helps to repair muscle tissues that were stressed during exercise. Some ways that we can improve or even reduce the epoch effect is firstly by a proper warm-up. So warm-ups, that set of exercises that we do before the main workout. The primary purpose of a warm-up is to prepare the body physically as well as mentally for exercise or before the main activity. So good warm-up typically includes like low intensity cardio exercises like jogging or cycling, some dynamic stretches and some sport-specific movements. For example, if you are playing soccer, you'd want to do some agility in your warm-up. Now, the benefits of the warm-up are increased blood flow. So warming up gradually increases your heart rate and the blood flow to the muscles, making sure that they receive oxygen and nutrients that they need to maintain that level. It improves muscle elasticity. So through warming up, we raise the temperature of the muscles, which makes them more flexible and less prone to injury. It enhances nervous system activation. So it helps to sort of prime the nervous system, which can improve coordination, reaction time, and performance. But the most important one for us is that we have a gradual increase in heart rate and breathing, and this helps to ease the body into exercise and reduces the sudden demand for oxygen and minimizes that initial oxygen debt. Active recovery is also important for repaying the oxygen debt or the epoch, and it's much better than complete rest. So after exercise, some sort of active recovery is very important. It's better to do some sort of active recovery than it is to just do complete rest because of a few things. Firstly, enhance blood circulation. So when we do a recovery, like a slow walk or a slow cycle, our muscles continue to contract. Even though it's at a lower intensity, we're still working those muscles, and that promotes better blood circulation, which helps deliver more oxygen to the muscles so that they remove the byproducts like lactic acid, which accumulate during exercise. It maintains our oxygen uptake, so it maintains an elevated level of oxygen consumption compared to complete rest, and that helps to sustain the body's ability to clear out the excess carbon dioxide and replenish the oxygen levels that were depleted during exercise. And then it also helps to restore muscle energy stores. Gentle activity during active recovery helps us to quickly replenish our ATP and our phosphocreatine stores in the muscles. So we want those stores. They're essential for muscle contraction and are depleted during intense exercise. And then finally, it reduces muscle stiffness. So with an active recovery, we can help to reduce muscle stiffness and muscle soreness, which is very common after intense exercise. And we've got this gentle movement that can help prevent the buildup of fluids and waste products in the muscles. You might have seen active recovery. The best example I see of it is in the Tour de France. After they've just finished cycling for however many hours, they get on a stationary bike and continue cycling to have all of these different benefits after exercise and help repay that oxygen debt or help repay the EPOC that they've created. Okay, the last concept that we're going to cover today is the lactate threshold. It's a really important concept in exercise physiology and sports science, particularly for endurance athletes. It's sometimes also called the anaerobic threshold. So lactate threshold, anaerobic threshold, you know that anaerobic glycolysis creates lactate, so it sort of means the same thing. So lactate threshold refers to the exercise intensity where lactate starts to accumulate in the bloodstream faster than it can be removed. And this point, this threshold, is often associated with a significant increase in fatigue and a shift in the body's metabolism. So if we can identify our lactate threshold, which we'll see on the next slide, if we are working below our lactate threshold, the body is using aerobic metabolism, which is more efficient and sustainable. We're able to do it for a longer period of time. If we're working above the lactate threshold, anaerobic metabolism is going to take over, leading to faster energy production, because we know that anaerobic metabolism is quicker than aerobic, but it also fatigues a lot quicker due to the lactate accumulation. So you can imagine already by now, if you can pinpoint where the lactate threshold is, you know, okay, I need to be below that for my body to work aerobically, for me to be able to sustain this exercise for a long period of time, or I need to work above that for anaerobic metabolism. So let's have a look at how we actually find the lactate threshold. So have a look at the graph first on this slide. So we've got blood lactate up the y-axis and power along the x-axis. So this person was likely on a stationary bike when they did this measurement. To measure blood lactate, it's very similar to measuring blood sugar or blood glucose. You sort of prick your finger with a little needle. That you might have done this before or seen someone do it. You have a little prick in your finger, it creates a little droplet of blood, and then you use a tiny little computer with a strip in it to sort of suck up that droplet of blood, and it gives us a blood lactate reading. So this person was on a stationary bike. As they're on the stationary bike, we're slowly increasing their power output, and we're measuring their lactate in their blood at each increase in power output. So they started at 100 watts and they finished at 300 watts. So we can plot what the blood lactate was on this graph and draw a curve. Now there's actually two lactate thresholds. There's lactate threshold 1 and lactate threshold 2. Generally, lactate threshold 1 is at around 2 millimoles of blood lactate, or millimeters of mercury of blood lactate. And you can see on the graph here that 2 millimeters of mercury occurs at about 160 watts. And then the second lactate threshold is normally at around 4 millimeters of mercury, and this occurs for this person at about 230 watts. You can also think of it as lactate threshold 1 is where we have this first sort of increase in the inflection of the curve, and then lactate threshold 2 is when the inflection of the curve increases even more. It's a very strong indicator of endurance performance, because I know that if I'm between lactate threshold 1 and lactate threshold 2, I'm not going to accumulate blood lactate that I can't continue. So if I'm working within this yellow zone, this threshold, I can probably maintain that for a while. If I was in a competition, let's say I was in a race, I'd probably want to be working right on lactate threshold 2, like right on the cusp of producing too much lactate if I really wanted to go hard and to win this race, right? But it's very interesting to know this. So if I was in a cycling race, say these were my numbers on this graph now, and I was in a cycling race, by doing this measurement, I know that I could probably do 230 watts for that race and won't sort of accumulate that muscle fatigue that occurs with the lactate byproduct of anaerobic metabolism. It's been suggested that the anaerobic or the lactate threshold is a stronger indicator of performance, endurance performance, than VO2 max. And that's because it's been seen before where athletes can have no increase in their VO2 max, no increase at all in the VO2 max, but they can improve their lactate threshold. And if they can improve their lactate threshold, it means they're able to sustain a higher output for a given period of time, for a sustained period of time. So lactate threshold 2 is the upper limit for endurance athletes. That's our threshold. Above this threshold, we have too much lactate, which makes our muscles turn acidic. It decreases the pH of the blood, and that's referred to as metabolic acidosis. So I want you to think about what it would look like in this graph if I was to do a lot of training trying to improve my lactate threshold. What would a better lactate threshold look like? What would the curve look like? Try and picture in your head. Would this curve move to the left or move to the right? Would it move up? Would it move down? And it would actually, hopefully you've all got this now, it would move to the right. So someone with a better lactate threshold would be this red line here, where that first inflection point occurs at around 220 watts, and the second inflection point occurs at around 275 watts. So this is what we'd want to see if we were trying to improve our lactate threshold. This now means that in the race, I can do 275 watts instead of 230. Okay, and then this relates somewhat to different training zones. So if you've got a Garmin or an Apple Watch or you use Strava, it might tell you your different training zones, and the lactate threshold is threshold training zone, normally referred to as training zone 4. And this is at lactate threshold, where the blood lactate is somewhere between 4 to 6 millimeters of mercury. So this is the uppermost point of that LT2, the second lactate threshold. It should correspond to about 87 to 92% of your maximum heart rate. And then we've got RPE values here. So this is rate of perceived exertion. There's two different types of RPEs you can take when you ask people on a scale how hard did that feel. You can show a scale that's 6 to 20, or you can show a scale that's 1 to 10. So if you show the 6 to 20 scale, it should be somewhere around 14 to 16. If you show the 1 to 10 scale, it should be somewhere between 5 to 6, and that should be hard. So it's hard to maintain the threshold, but we still don't have that accumulation of blood lactate that might lead to fatigue. So you can refer back to this table when it comes time to do your assignments or your IA, but it's a good table to have and to be able to sort of understand the different zones and where they might lie in terms of blood lactate and in terms of heart rate percentages as well.